All right, this is the second recording for the cell. So now we're going to talk about the cell membrane. The cell membrane is going to be key here in anatomy and physiology. And that is because your cell membrane is going to determine everything that can easily exit or enter the cell. And it's going to determine what easily enters and exits the cell. Things like ions, smaller molecules, polar molecules, nonpolar molecules, gases, water, etc. Right. So your cell membrane, which is also commonly called plasma membrane, so you'll see that name as well. Sorry, that was very ugly written. Plasma membrane. Okay. Your plasma membrane will be the gatekeeper, as I mentioned, determining what can enter and exit the cell. But it is also going to have a lot of additional things on it. It's going to have receptors that serve to determine or to say, I am who I am. There are many white blood cells, for instance, that'll run through your body trying to destroy things that shouldn't be there, like bacteria and viruses and cancer cells and worn out cells that are no longer functioning. And so therefore, those white blood cells need to know what is or isn't functioning, what is part of the human body and what shouldn't be within the human body. And so those cell surface receptors can help tell those white blood cells, hey, no, I'm a liver cell, don't kill me. Or, hey, I'm like just not working properly, please do get rid of me. Or, oh, gee, look, that bacteria doesn't belong here. That virus doesn't belong here. Let's kill it and get rid of it. The other thing, a reason why you're going to have receptors, or an important reason why you're going to have receptors on your plasma membrane, is because they're going to help receive signals. So if one part of your body is sending a signal to another part of your body, then those cell surface receptors can receive those messages. We're going to look at that in a little bit more detail. A third key important function of cells is going to be, or I should say of the plasma membrane of cells, is going to be to hold cells together. So there are some cells in your body that are going to be free-flowing, which means they're not necessarily attached to other cells. An example of that would be your red blood cells. Red blood cells and white blood cells are going to run through the bloodstreams, just kind of hanging out and going from one side to the other, doing their different purposes and functions. But cells that need to line organs are going to need to form tight structures. So they're going to need to be attached to each other to make a lining. And so this would be an example of many different cells attached to each other to form a lining. And in order for those cells to hold on to each other, the cell membrane of each cell needs to form a junction with the other cell. So cell junctions will also be an important function. Great. So let's start by understanding that your plasma membrane, your cell membrane, is going to be made out of something called phospholipids, okay? Something very important called phospholipids. So if you remember, there are 4 types of macromolecules: proteins, carbohydrates, lipids, and nucleic acids. Your lipids, if you remember, are something called hydrophobic, which means that they fear water. They do not like water. They will not dissolve easily in water. They do not mix well in water. Right? So your lipids are hydrophobic. The exception to this guy is going to be your phospholipid. And that's because your phospholipid is going to be composed of 2 parts, a head and a tail. The tail is, like any good lipid, hydrophobic. Does not mix with water. However, the head is actually hydrophilic, which means that it does mix with water. Okay. And this is going to be important because we're going to see how the plasma membrane will form a nice little bilayer due to the hydrophilic and hydrophobic functions of the phospholipids. So let's scroll down here to the phospholipid bilayer. You can see here that the bilayer looks like this guy here. So because these heads are hydrophilic and the tails are hydrophobic, they're always going to situate themselves with the heads facing watery areas and the tails facing away from the watery areas. Okay, so when we look at a image of a plasma membrane, you can see here how this is the exterior of the cell, and the exterior of the cell contains a lot of water with many other substances. And the interior of the cell over here also contains water with a lot of substances. So here's your cytoplasm, and up here is the extracellular fluid. So because the tails of the phospholipids are hydrophobic, they will naturally move away from those exterior and interior surfaces, and then they're going to face each other, while the heads will stay facing the interior and exterior sides of the cell. And this will then form a phospholipid bilayer where the interior of the membrane then forms a fluidity, allowing things to move through there, especially things that will be hydrophobic, not liking water, right? So that's what you're going to have there. Cholesterol can be found hanging inside of this. If you remember, cholesterol is a type of lipid, which means it's hydrophobic. Cholesterol is found hanging out inside the membrane because cholesterol helps determine how fluid that membrane will be to obtain its correct function. So what kind of things can be able to pass easily through this membrane? Well, here is where you're going to have to think about molecules that we've already talked about, and what is polar and nonpolar, and what is hydrophobic and hydrophilic. So if we're going to talk about things easily moving through the membrane, you have to first of all think, what could easily go like this, in through this membrane to get inside the cell, or easily out through this membrane to get outside of the cell? Well, for starters, it's going to have to be a small molecule, right? So small molecules are key to passing freely through the plasma membrane. The other thing is, because the interior here is hydrophobic, if you are a hydrophilic or polar molecule, then what's going to happen? You'll start coming this way, and then you won't be able to get through because this area here will push you out. So polar molecules can't easily pass through the membrane, but nonpolar, so small nonpolar molecules will be able to pass easily or freely through the plasma membrane, right? So things like gases, for instance, will be able to freely pass through the plasma membrane. So oxygen, carbon dioxide, and other nonpolar molecules that you can think of will easily pass through as long as they're small enough. For everything else, you're going to have to use something to be able to get through the membrane. And so here is where these proteins that you see crossing the membrane come into play. So you guys see this little one here? That there is a transmembrane protein because it crosses through the membrane. It crosses through that entire plasma membrane. So if I am a larger nonpolar molecule, or even if I am a polar molecule, be it small or large, since I can't go this way, instead I can go through here to this transmembrane protein to get in or out of the cell. Right? And there are going to be many different types of transmembrane proteins, and we're going to look at those and what they are. Other things that you're going to notice here on the plasma membrane are going to be things like, let me change color here. Let me use the red. This guy here, this is a peripheral protein right here, and we've got another peripheral protein here. Peripheral proteins have different functions within the cell. For instance, they can help hold the cytoskeleton to the plasma membrane to keep the plasma membrane in place instead of just like falling off of the cell membrane. So there's many different functions for them. You will always notice that they're going to be to the side. The peripheral means along the side. So peripheral proteins are going to be on the side. Transmembrane proteins will be crossing, and then we're going to have things like your glycolipids and your glycoproteins. Let me erase some of the stuff here that you're going to be seeing. So here is a glycocalyx, here's a glycolipid, and then you're going to have different types of those around. So these are part carbohydrate and part lipid, or part carbohydrate and part protein. And they're going to have many different functions, of which we're going to look at soon. Okay, so here are different functions of the membrane proteins. So of the proteins that you can find along the membrane. You can transport substances, like I mentioned before. So substances that can't normally or freely pass through the membrane can then more easily pass through it using these transporting substances or transmembrane proteins. Some can act as receptors, so they're going to receive messages and then allow those messages to enter into the cell. Some will work as enzymes, which is to speed up processes and do things like help break things down or help make things. Some will function as peripheral proteins, like I mentioned before, to hold the cytoskeleton in place. So that way, or I should say, hold the plasma membrane to the cytoskeleton to hold the plasma membrane in place so it does not fly off of the cell, of the rest of the cell, okay? And so that's structural support there. And then we have things like making those cell junctions. Remember when I talked about sometimes you need to have many cells together to form, for instance, a lining of a surface or of an organ? Then all of those cells need to be held together, plasma membrane to plasma membrane, and they use these kind of proteins that are located on the plasma membrane of adjacent cells to link to each other, and they're called linking adjacent cells. Makes perfect sense, right? Oh, by the way, a lot of these guys that you see here that act as receptors, they're going to have a combination of transmembrane proteins with glycoproteins to help receive messages, or glycolipids. Okay, so let's talk a little bit about how some drugs, for instance, can bind to the membrane receptor. So as I mentioned before, some of those proteins that are found in the plasma membrane are going to serve to receive messages and then to be able to transmit those messages inside the cell. Okay, so drugs can do that. They can bind to certain cell surface receptors on the plasma membrane, right? And if what they can do is they can either mimic the normal action of something that the cell would receive, or it can work against it. So if I have a cell membrane and it has certain receptors along it, right? This is a double membrane. It's got certain receptors along it to receive messages. So for instance, receiving a message like dopamine through this cell. But I can have certain drugs that are going to either work as agonists, mimicking or stimulating that receptor. So cocaine would be an example of a drug that is an agonist because it stimulates the receptor so that dopamine is received more strongly or more continuously and keeps giving you those euphoric symptoms that accompany cocaine use. Of course, that's going to have a lot of not good secondary effects. So one of the things we're going to do is later in the semester, we'll be looking at the nervous system. And when we get into the nervous system, we're going to do a really fun activity called Mouse Party. And in that one, you're going to see how mice react when they're put on certain drugs like cocaine and heroin, meth, marijuana, LSD, etc. Don't worry, they're not real mice. It's just a simulation. And you'll see what happens in the brain and in these cell surface receptors, where they will either form agonist or they can form antagonist to block the receptors, okay? A good example of an antagonist are going to be your antihistamines. So antihistamines, what they're going to do is they're going to block the receptor from receiving the message, from receiving an allergic response. So if you are allergic to, say, pollen, tree pollen, like I am, then what's going to happen is the antihistamine is going to block the receptor for that histamine, because your body will produce histamine in response to pollen entering through the nasal cavity. And you take an antihistamine, and what it does is that plasma receptor, naturally the histamine will go like this, but this antagonist is going to form a little block and stop that histamine from being able to get in. So you have different types there. All right. And again, we'll be looking at those more detail when we get into the nervous system. All right, so the next thing we're going to talk about is how things actually enter and exit the cell, and how they may utilize or not utilize energy to perform those movements. So I'm going to pause here to not make the video too long. And in the third video, we're going to look at movement throughout the cell membrane.